All right, well, welcome to the stream, the show. And let me see if it works. Okay, so that worked, but the audio was probably didn't show up for you guys. Let's see. So, I think that didn't play. So let's see again. Hello? Yeah, it doesn't play. Okay, so let's work on this a little bit. You know, stay with me a little bit. Come on. Um, let's see. All right. Hello. Now let's try this. Let's see how this works. Nope. Nope. What about this? What about this? About this? Oh, I see. Oh, I see what I need to do. First of all, let's take this down. Boom. Boom. Here we go. Let's try this now. This is gonna work, I'll bet. Let's see. Hello, everyone. Oh, wait, that's not gonna work. Let's stop.
Welcome to everybody. My name is Jeffrey West, and I'm
There we
on the
go.
faculty of the Santa Fe Institute, and it's my real honor and distinction to be
There
able to
we go.
introduce our wonderful speaker tonight.
Okay, that was gonna be Suskind. Okay, so all right, everybody. Let's start this video for real. How long has it been so far? Oh, I don't know. It doesn't say. Oh wait, I could find out this way. I could find out. Boom on this guy. Live streaming. 4 minutes. Sorry about that. 4 minutes. Okay. So the subject is mind viruses. This is a metaphor. So first of all, I'm gonna have to, because you just like, virus is a germ that infects me. You guys don't get it. So we're gonna have to go through this step by step. Okay, first of all, DNA. That's the twirly thing that you're familiar with. That is when the data is stored. It's stored in a chemically stable way. Now, when the DNA is used, what happens is a protein comes in and strips off a copy of half of it. So the RNA is that half. You can certainly see how that is a half, right? So it's a string by which, because these things all match in here, you can figure out what the other half is always from it. The yellow will be red, and so something's going to come along and, ding, 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 match it, and that's going to cause a protein to be created. All right, so DNA packaging is the first thing we're going to talk about. The fact that DNA, like a DNA molecule, is like 3 feet long, but it gets packed into a tiny little ball that can still be accessed like a library. We're going to look at that. We're going to look at the nuclear import and export, so things that go in and go out of that. Then we're going to look at how the little bits of RNA that are copied out of segments of the DNA, the half part, right? Uh-oh, what's that? Can you hear that? That was terrible. Okay, and then we're going to look at how that creates amino acid string, which is folded into a protein, which is a little robot or a tool, right? And this is all to work towards what the metaphor is. So in order to understand the metaphor of a mind virus concept, you need to understand how real viruses work, which requires understanding how cells work. Okay, so what we're going to do is I have some videos. Okay, here we go. They're in my temp directory. First of all,
in this animation, we'll see
how
the
DNA
remarkable
is
way our
packaged.
DNA
So that's
is
DNA.
tightly packed up to fit into the nucleus of every cell.
So
The
3
process
feet
starts with
long.
assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small
6
that
feet.
10,000
2
nuclei could fit
meters.
on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
All right, so you pack the DNA into a little compact structure that is able to act like a little library. So what would be 6 feet long is inside a little tiny cell, and it can be actually entered as like a library. Nuclear import and export.
The nuclear pore complexes are the only channels through which small polar molecules, ions, and macromolecules can travel between the nucleus and the cytoplasm. By controlling this traffic, the nuclear pore complex plays a fundamental role in the physiology of all eukaryotic cells. Most proteins and RNAs are too large to move through the complex by passive diffusion and must move by an active process in which appropriate proteins and RNAs are recognized and selectively transported in a specific direction. Let's consider proteins imported from the cytoplasm to the nucleus. Only a subset of proteins can enter the nucleus, and they are allowed in by virtue of having specific amino acid sequences called nuclear localization signals. Nuclear localization signals typically consist of one or two short segments of basic amino acids. Nuclear localization signals are recognized by nuclear transport receptors called importins, which carry the cargo proteins into the nucleus. With its cargo attached, importin binds to specific nuclear pore proteins in the cytoplasmic filaments. By sequential binding to more interior nuclear pore proteins, the complex is translocated through the nuclear pore.
So it's, it has a key that allows entry, and then it goes into the DNA library, and it can import DNA sequences and also export them. And then when it exports them as RNA, and which drives this, copying and production that we'll see in the next video.
At the nuclear side of the pore, the complex is disrupted by the binding of a protein called RAN to importin. RAN carries a molecule of GTP. This binding changes the conformation of importin, which then releases its cargo protein into the nucleus. The importin-RAN complex is then re-exported through the nuclear pore. A protein in the cytoplasm called RAN-GAP, for GTPase activating protein, stimulates RAN to hydrolyze its GTP to GDP, an action that triggers RAN to release importin back into the cytoplasm. RAN plays a key role in protein import and export. Note that while RAN is bound to GDP, it cannot disrupt the binding of importin to a cargo protein. In an action that prevents the depletion of RAN from the nucleus, the RAN GDP formed in the cytoplasm is transported back to the nucleus by its own import receptor, a protein called NTF2. In the nucleus, another protein called RAN-GEF, for guanine nucleotide exchange factor, stimulates RAN-GDP to release its GDP and pick up GTP. In this form, RAN-GTP can disrupt the binding of importin and its cargo, triggering the release of the cargo in the nucleus. By the actions of RAN-GEF, found only in the nucleus, and RAN-GAP, found only in the cytoplasm, a steep gradient of RAN-GTP and RAN-GDP is maintained across the nuclear membrane, with RAN-GTP inside the nucleus and RAN-GDP in the cytoplasm. Similar to imported proteins, proteins are targeted for export from the nucleus by specific amino acid sequences called nuclear export signals. Nuclear export signals are recognized by receptors within the nucleus called exportins, which direct protein transport through the nuclear pore complex to the cytoplasm. RAN-GTP promotes the formation of stable complexes between exportins and their cargo proteins. Note that this same form of RAN does the opposite for importins and their cargoes. The effect of RAN-GTP binding on exportins dictates the movement of proteins containing nuclear export signals from the nucleus to the cytoplasm.
Which
Following
is
transport
RNA,
to the
right?
cytosolic
It's
side of the
exporting
nuclear envelope,
RNA.
GTP hydrolysis and release of RAN-GDP leads to dissociation of the cargo protein, which is released into the cytoplasm. Exportins are then recycled through the nuclear pore complex for reuse.
All right. Now the next thing is mRNA translation. So what's exported from the nucleus is a little genetic fragment, right? A particular idea in that's stored in the worldview that is the DNA. So let's look at that.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several
Protein's
million
like a
ribosomes
behavior.
in a
It's a
typical
little robot.
eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger.
So basically, you have a little bit of RNA. It gets taken out of the nucleus. In a healthy mind, it's processed by these ribosomes that run the program that is a part of that is the RNA. Runs that program. That program basically is a printer. It prints the amino acids, which is a string of chemicals. Then in the water that's in the cell, the cytoplasm, it gets folded into a little balloon animal that's super complex, and it's a little robot. It can be a motor protein with legs that go along. It can be a vesicle. It could also just be an antibody that's like a little, I don't know, a scissor or a cutter that just floats around, and like the enzymes in your saliva just chops up molecules, like because of its shape. But it can also be like a little robot that's walking along, a motor protein that's walking along um, the cytoskeleton.
RNA, which then passes through the ribosome like a computer tape.
Mm-hmm.
The amino acid building blocks, that's the small glowing red molecule.
Now here's the thing they don't really know, but how does the ribosome get all these amino acids? Well, the idea in randomness of life is that, oh, there's so many, they keep bumping along, and they just click, 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 the ones that fit and the ones that don't bounce off. That's the idea. But basically, this is all organized by the water molecules inside the cell. Highly packed water molecules that I believe are probably doing a computational function of some sort. But it doesn't really matter. Maybe it's just that the water flow evolved inside a cell to be very stochastically intense, so that there's always bump, 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 bump. There's always enough of a certain kind of randomly presented amino acid to like set and program into this change, which is controlled by the proteins that are the ribosomes.
Are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, The tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site, and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides, or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
All right, so now the last thing is that the protein has to be folded into the little machine. So after it gets transcribed, it's a long string of amino acids, and it has to be folded into a protein. Now that last video showed it being folded into a protein as it got excreted from the copy machine. So I'm not sure if that's a certain thing or not, because a lot of times in the past I've read that the amino acids come out as string and then nobody knows how it folds. Why does it fold this way versus that? I figure it's because the water is organized is arranged in an organized way, so it's performing these active computations that fold the protein. But it could just be driving the ribosome, and maybe it does come out folded as a result. This is the last video, you guys. It's only 46 seconds. See, it's showing it being folded as it comes out, I think. Yep. Okay. All right. And then the proteins are little robots, motor proteins and things like that. I mean, I could show a motor protein, but you know what I'm talking about. There's a lot of them that have legs and are doing things, and other ones are just like a shape that if it wiggles, chops up another protein so you can digest it, right? All right, so in the healthy mind, now we're getting into the metaphor. In a healthy mind, the RNA that comes out of the nucleus is like an idea in the mind. So why is that? Because the RNA is used to build a protein, and the protein does some activity in the cell. Well, an idea is used to build an idea, an active idea, and an action, something in the mind that you then believe or act on. So when you have an idea that comes out of your worldview, right? You take your worldview is all of these views. Maybe something's happening, like a dangerous situation. Your mind can send certain, quasi-analogous proteins into your worldview, bring out ideas, and then in reaction to the stimuli, generate behaviors like fight, flight, or whatever is going to be generated. In the RNA, in the cell, it's generating a protein, and these proteins are internal actions, like to generate a certain protein. Some proteins are antibodies. So you might have a stimulus that um, causes an RNA fragment to come out, be chosen, and then that RNA fragment builds a protein that's an antibody. All right. So here's how the mind virus is similar to a regular virus. RNA is like an idea. When the RNA is in a cell, the cell runs that idea as if it was a program that got booted up or loaded. An idea, when it goes in the mind, is run also like a program that was loaded. You get an idea, if you accept it into your mind, then it in turn generates ideas. So the idea creates other ideas, and those ideas are behaviors, ideas that you act on. Some of them might, you act on a little bit or even not at all, in other words, a lot. Similarly, when a virus injects RNA, it creates virus proteins that do things the virus wants. Now, those virus proteins could be complete virus capsules, but they can also be other more subtle things that cause the cell to do side effects besides creating more of the virus. Of course, one thing that's always going to be done is that the virus is going to be recreated. And in the analogy, an idea is such that it's going to create behaviors, but those behaviors are going to be designed to recreate the idea, both inside the mind and outside the mind. Because it's inevitable when a virus takes over the programming of cell, it's going to create all these proteins, and at least many of them are going to be the virus itself, encapsulated in seed form, sent out into the world. Either the cell dies, the virus goes in there, causes a lot of the virus to be created, and the cell dies, and the virus, escapes because the cell evaporates and deteriorates after it dies. Or if it's a more subtle virus, because it seems like every level of subtlety is available, then, it might it might create copies of the virus that get excreted by the cell, and the cell keeps going fine, so it can create more of them. In the analogy, you get an idea from the outside world, and it creates a behavior. That behavior creates the representation of ideas in the real world, and ideas that you have, and also internal behaviors, side effect behaviors. And so this is a picture of how this happens. People get ideas from outside that replicate themselves inside the brain, that then generate and excrete ideas into the world. And this is something all of you insane people need to, think about. That's all I'm saying. All right. Nobody's online, right? No, I'm always going on the Discord. So I decided to do the Discord description at the end. So this is a text channel on the Discord up there that says broadcasting text. And then as you can see, am listed over there. That's where if people were in the audio, they would be able to chat in this video right now that's being streamed. This HTTPS location, that's a Discord invite. If you were to type that in to your browser, it would get you into my Illusions Exist channel. Down in the description is one you could click on. I assume that one is going to run out sooner than this because it's not infinite, but it's very many invites that you can use to get in. So like when this one runs out, that one will probably still be available, and you could do that to get in. And then if you go into the broadcasting text or the broadcasting audio channel that's just titled broadcasting, you would be in my show. surely see how awesome that would be for you. All right, everybody. This was my description of the mind virus and why it's a very good metaphor. And I'm sure everybody accepts it. Plus, there's the Euler identity down there in the bottom. How is that not great? Cheers, everybody. And I hope you do well in your life and everything else that might be going on and stuff like that. Cheers.